Esta es Arturo Abundes Corona. Arturo Abundes viene desde la Ciudad de México, es médico cirujano, graduado de la Universidad Nacional Autónoma de México, neurólogo certificado por el Consejo Mexicano de Neurología y tiene especialidad obviamente en trastornos del movimiento y enfermedades neurodegenerativas por el mismo Instituto Nacional de Neurología y Neurocirugía. Es también investigador y profesor adjunto del Laboratorio Clínico de Enfermedades Neurodegenerativas de la UNAM y del Instituto Nacional de Neurología y Neurocirugía, y está adscrito también al Centro Médico ABC en Ciudad de México. Así que Arturo nos va a presentar de forma similar a como Mario lo hizo, una descripción de cuál es la situación de la enfermedad en México. Arturo, bienvenido.
Muchas gracias. Hola, listo, perfecto. Pues muchísimas gracias y sobre todo gracias a Factor H por la invitación. Es un honor para mí estar aquí, estoy muy contento de representar a mi país aquí en Colombia. Y bueno, les quiero platicar un poquito sobre generalidades, es una breve reseña sobre cómo es la situación de Huntington en México actualmente. Vamos a empezar primero con un poco de contexto sobre mi país, esto para que entendamos por qué la situación de México es tan compleja en este momento. México es un país muy grande, es el 13º país más grande por área, es el país 10º más poblado del mundo y es el país con población habla hispana más poblado también del mundo, y es la economía número 15 por Producto Interno Bruto y la número 11 por poder de adquisición. Esto hace que México sea un país étnicamente y culturalmente sumamente diverso. Tenemos una historia sumamente rica. Básicamente es esta mezcla entre las poblaciones nativas previamente y la colonización europea que vino hace más de 2 siglos, y podemos observar que la composición genética de nuestro país es sumamente diversa. Más o menos 60% de la población se define como mestiza, básicamente una mezcla entre españoles e indígenas. Tenemos un 20% que es predominantemente blanca y tenemos 15% que es solamente indígena. Más o menos el 3% es afro-mexicano y tenemos un porcentaje menor de 2% aproximadamente de otras etnias. Y como había mencionado, principalmente tenemos una población americana mezclada con europea, y esto hace que la población sea genéticamente heterogénea. Tenemos algunos clústeres de población mexicano-africana que no es tan frecuente en México como otros países caribeños o de Sudamérica, pero ahí está bien, ¿está mejor ahí? Listo, ustedes me van diciendo. México también está pasando por una transición demográfica muy importante. Hace unas cuantas décadas teníamos una pirámide en donde la base era sumamente amplia y teníamos una población envejecida muy pobre. Ahora vemos que esto se está normalizando un poco y estamos en esto que se llama inversión demográfica, en donde tenemos una inversión gradual y lenta de la población demográfica, la pirámide demográfica, y esto hace que tengamos una carga muy importante de enfermedades neurodegenerativas que generan y degenerativas en términos generales, que generan una carga para salud pública, para los sistemas muy importante, pero dado que es un país sumamente desigual en términos económicos y sociales, tenemos una carga también de enfermedades asociadas a la pobreza muy importante. Uno de los grandes problemas de salud en México es que la salud, sobre todo la salud especializada, y estamos hablando de la salud especializada en neurología o de cualquier otra especialidad, no se diga de trastornos del movimiento, está centralizada en las grandes metrópolis. Básicamente tenemos centros de atención especializada en Ciudad de México, Monterrey, Guadalajara y quizás algunas otras ciudades también mayores, pero básicamente esto hace que la gente que vive en zonas rurales o en zonas más remotas tengan que viajar a las grandes urbes para recibir atención especializada. Prácticamente no hay neurólogos especialistas en trastornos del movimiento o grupos multidisciplinarios de Huntington en áreas rurales del país. Y esto implica que los pacientes además gasten una cantidad importante de dinero para viajar a atenderse en los centros urbanos. Otro problema es que el sistema de México está dividido. Tenemos un sistema de salud público mixto con además algunas instituciones privadas, pero dentro del sistema público no hay una unificación de los 8 sistemas de salud que tenemos. Entonces tenemos 8 instituciones públicas distintas, cada una con sus reglas y cada una con beneficiarios específicos. Por ejemplo, tenemos el IMSS, que es básicamente la seguridad social para cualquiera que tiene un trabajo registrado en el gobierno, tenemos el ISSSTE, que es para aquellos trabajadores del Estado, y tenemos otras instancias más pequeñas que cubren un porcentaje importante de la población en México. Pasando un poquito del pequeño brevario que les dije sobre México, quiero hablar acerca de qué datos tenemos en México sobre la enfermedad de Huntington. En realidad no tenemos datos epidemiológicos y no sabemos cuál es la prevalencia real en nuestro país. Sabemos que se asemeja a lo que está reportado a nivel mundial, pero no tenemos un estudio epidemiológico que nos diga exactamente cómo se comporta la enfermedad en México, lo cual sería muy interesante dada la característica genética y étnica de nuestra población. Lo que sabemos es que desde los 80s se definieron dos áreas en el norte del país, específicamente en el estado de Tamaulipas, que podrían considerarse clústeres genéticos de alta prevalencia de la enfermedad. Son poblaciones pequeñas, en realidad estamos hablando de estos municipios de Tula y el municipio de Casas, cada uno con una población, uno de 10,000 habitantes y otro con 4,134 habitantes. Y posterior en los 90 se trató de hacer una expansión sobre los datos epidemiológicos en estas regiones, pero por un tema más bien social y de inseguridad asociado al narcotráfico, no hemos podido acceder a estas regiones probablemente con mayor prevalencia. Y esto sucede en grandes partes de México rural. En las últimas 2 décadas hemos tenido muchos problemas de seguridad para viajar a zonas rurales y esto hace que no tengamos datos epidemiológicos en comunidades específicas. De esta manera, dado que no tenemos un sistema epidemiológico o datos epidemiológicos de la prevalencia en México, los datos que tenemos sobre Huntington en el país se basan en los centros específicos que tratan estas enfermedades. Voy a hablar yo específicamente de los datos que tenemos en el centro en donde yo trabajo, que es el Instituto Nacional de Neurología en la Ciudad de México. Aquí, desde 1990 hasta ahora, tenemos un buen registro bastante confiable de pacientes con Huntington, en donde tenemos 625 pacientes con Huntington que han sido captadas y estudiadas. Se han realizado más o menos 1,700 pruebas genéticas desde entonces, de las cuales 1,250 han sido positivas para el gen de la huntingtina, para repetidos de CAG, y tenemos de estos más o menos 175 con enfermedad premanifiesta que han sido detectados. El restante, 450 pacientes que salieron negativos, tienen otra serie de enfermedades que pueden ser fenocopias de Huntington, como Huntington-like tipo 2, algunas ESCAS, tenemos también enfermedades por depósito de hierro, algunos casos de neuroancatocitosis y tenemos 2 o 3 casos por ahí con estos expandidos de C9orf72. Realmente esto no ha sido publicado, pero esto es lo que nosotros tenemos en la consulta durante este año en la Clínica de Trastornos del Movimiento, en donde yo trabajo. Hemos visto hasta el día de hoy, del 1 de enero del 2022 al día de hoy, 102 pacientes con enfermedad de Huntington. Tenemos 54 mujeres, 48 hombres, de los cuales 44,6% son casados, 26,1% solamente completaron la educación primaria, que esto es también un problema frecuente en México, y solamente 17,4% tienen una educación profesional. 74,3% no tienen empleo, lo cual es importante mencionar, dado los costos tan elevados que los pacientes tienen que tener para el tratamiento y también para los traslados a los centros de hospital y en general cualquier otro cuidado que requieran ellos. De esta población que estudiamos, la edad mediana es de 37 años, es una población relativamente joven, aunque tenemos algunos pacientes mayores, 86 años. La edad de inicio de los síntomas motores promedio en estos pacientes fue de 39 años y la edad de inicio de los síntomas neuropsiquiátricos o el primer síntoma neuropsiquiátrico es de 41,4 años, con el diagnóstico molecular realizado a los 46,3 años. Esto también indica de que hay aproximadamente 6 o 7 años de retraso en el diagnóstico desde que aparecen los síntomas motores a que se hace la prueba molecular. En términos de la carga motora de la enfermedad, 82,7% tienen una historia positiva para enfermedad de Huntington, 2,5% se presentaron como Huntington juvenil, tenemos 89,7% que básicamente tenían corea como el síntoma predominante, 9% tenían un temblor dominante y 2,6% tenían distonía, y esto corresponde a pacientes obviamente más jóvenes. En términos de la carga neuropsiquiátrica de nuestros pacientes, 38,6% tenían síntomas depresivos mayores, esto medido por la escala de Hamilton de depresión, 35,7% irritabilidad, 37,1% tenía algún tipo de deterioro cognitivo leve y 14,4% ya cumplían criterios para decir que tenían demencia. Y 11,4% tenían ya ideación suicida, que también ha sido importante mencionar. En términos de cómo tratamos a los pacientes en el centro, y esto tiene mucho que ver con qué tan accesibles son los medicamentos para nuestros pacientes, porque en realidad ellos tienen que costear parte de los medicamentos, no les cubre por completo la seguridad social. El 66.6% estaban tratados con antipsicóticos atípicos, sobre todo olanzapina. Teníamos 16.6% con tratamientos con antipsicóticos típicos, tetrabenazina 20.8%, esto por un grupo de fármacos que llegaron a través de una fundación y que tuvimos ahí esto, pero esto no es realmente tan real. Si nosotros vamos a años anteriores, el porcentaje de gente que tiene algún inhibidor de BMAT2 es mucho menor. Tenemos 50% de los pacientes con un inhibidor selectivo de recaptura de serotonina y benzodiazepinas en 21%, y tenemos un porcentaje relativamente importante de pacientes que no reciben ningún tratamiento farmacológico. Ahora, ¿cuál es el camino que un paciente con enfermedad de Huntington o con una corea, con diagnóstico o con una sospecha de enfermedad de Huntington tiene que cumplir para que nosotros hagamos el diagnóstico en nuestro instituto? Bueno, primero llega el paciente referido por un médico de cualquier otro lugar del país, llega al centro de referencia, que es el Instituto de Neurología, es evaluado por un neurólogo general y el neurólogo general lo refiere a la clínica de trastornos del movimiento y a neurogenética. Nosotros hacemos ahí la evaluación genética, la evaluación de diagnóstico inicial, esto incluye a veces resonancia magnética, en casos muy particulares algún estudio molecular como el PET, y de aquí nosotros también hacemos una evaluación neuropsiquiátrica y neuropsicológica, que son fundamentales antes de nosotros poder hacer o dar el diagnóstico molecular. Y bueno, se hace la prueba molecular y empezamos el tratamiento sintomático. Esta es la batería de estudios que nosotros hacemos a todos los pacientes que entran a la clínica de trastornos del movimiento en México para su evaluación y su subsecuente seguimiento. Esto es parte de un registro nuevo que estamos haciendo y la idea un poquito es homogeneizar la evaluación de los pacientes desde el punto de vista clínico. Entonces, evidentemente se tiene que hacer todo el consentimiento informado con todo lo que esto implica para la prueba genética, se hace una evaluación motora compleja con las escalas habituales que ya sabemos cuáles son, y también se hace una evaluación del comportamiento y de la cognición con una batería neuropsicológica y con una evaluación neuropsiquiátrica bastante extensa. Algunos pacientes también y sus familiares se les somete a una escala que se llama SARIT, que es un cuestionario que tiene que ver con la carga del cuidador de estos pacientes, que nos parece sumamente relevante y de donde estamos obteniendo de un estudio que estamos ahorita desarrollando datos muy, muy interesantes. Ahora, en el instituto funciona a través de un estudio socioeconómico. Todos los pacientes que ingresan son sometidos a un estudio socioeconómico. La gran mayoría de nuestros pacientes están entre nivel 1 y nivel 2, pues son pacientes habitualmente en condiciones precarias socioeconómicas. Aquí la prueba genética para hacer el diagnóstico molecular cuesta más o menos $1. Esto es para pacientes que tienen nivel 1, pero aquellos pacientes que tienen nivel 5, que es el más alto, podría costar hasta $95. El resultado genético, una vez que se hace, es dado por un neurogenetista y también esto se hace a través del consejo genético. Uno de los grandes problemas que tenemos es que actualmente no tenemos guías de prácticas clínicas estandarizadas en México y por lo tanto dependemos de las guías internacionales, dependemos de las guías americanas, de las guías europeas, que es en donde nosotros nos basamos para tomar decisiones clínicas, pero el problema es que como no tenemos guías locales no podemos pedirle al gobierno que nos dé o nos cubra ciertos medicamentos, y esto es un gran reto con el cual estamos luchando todo el tiempo. La COFEPRIS, que es el órgano que controla los medicamentos aprobados para X o Y enfermedad en México, ha aprobado desde el 2013 tetrabenazina como uno de los agentes, digamos, modificadores de dopamina para Huntington, y dentro de los modificadores asociados a glutamato tenemos amantadina y riluzol. En realidad, la COFEPRIS, para la indicación específica de Huntington en México, no ha aprobado antipsicóticos, lo cual es muy sorprendente y se ha hecho un esfuerzo como para convencer a los pacientes, más bien a la COFEPRIS, que pueda hacer este cambio. Entonces, muchos de los pacientes que tienen que tener algún antipsicótico tienen de repente en el expediente algún diagnóstico adicional, además de Huntington, para justificar el uso de estos antipsicóticos. Es un tema absolutamente administrativo que no debería tener, no tiene sentido y debería ser algo más sencillo. En realidad, prácticamente todos los tratamientos actuales para Huntington están disponibles en México, inclusive tenemos algunos pacientes que lo gastan ellos de su bolsillo, pues son pacientes muy adinerados que tienen deutetrabenazina, etcétera, que lo importan de Estados Unidos. Se pueden conseguir los medicamentos y en el mercado mexicano tenemos el resto de los medicamentos sin tema. El problema es que la seguridad social cubre muy poca parte de estos medicamentos y esto implica que los pacientes tengan un gasto de bolsillo sumamente elevado. Esto no nada es para el paciente individual, sabemos que es un problema familiar. Y recientemente en el instituto, junto con el Seguro Popular, que es una instancia pública nueva, se abrió una farmacia en donde tenemos prácticamente una cantidad considerable de antipsicóticos y otros medicamentos que les podemos dar a los pacientes solamente al Instituto de Neurología de forma gratuita, y tenemos una cantidad limitada de tetrabenazina que hemos utilizado este año con bastante éxito. ¿Cuál es el rol de las asociaciones de Huntington en México, las asociaciones no gubernamentales? Es importantísimo. Entonces tenemos por aquí, por ejemplo, la Asociación Mexicana de Enfermedad de Huntington, que ha hecho esfuerzos tremendos para ayudar a las familias y a los pacientes individuales, en donde, bueno, ya nos habían platicado el día de ayer un poquito de lo magnífico que es este centro. Honestamente creo que es un centro referente para Latinoamérica. Aquí en la asociación tienen una serie de actividades diurnas importantes, tienen comedor, tienen actividades sociales, tienen visitas a casa, atención multidisciplinaria, y bueno, la intención es ahora tener mucho más relación con estas asociaciones no gubernamentales desde las instituciones de salud pública para poder hacer mancuerna y poder ayudar de forma más integral a los pacientes. En términos de los datos locales que tenemos, quizás estos son los dos estudios más importantes que recomiendo que ustedes, si les interesa, puedan adquirir. Tenemos estos estudios en donde, bueno, tenemos una cantidad importante de pacientes que han sido seguidos por el tiempo. El primer estudio no es tan reciente, es del 2009, pero bueno, tiene una población bastante grande de pacientes que han sido estudiados con diagnóstico molecular. Y por otro lado, tenemos este otro estudio sobre el diagnóstico presintomático y la importancia que los clínicos le damos al diagnóstico presintomático. Les invito a que revisen estos estudios. Yo los tengo por si alguien los quiere, los puedo compartir con mucho gusto. ¿Tenemos algún tipo de registro local de pacientes? En realidad todavía no está muy establecido, pero ahora en la Academia Mexicana de Neurología estamos trabajando en hacer un registro de Huntington muy parecido al REMEPARC, que ha sido un registro para Parkinson muy exitoso en el país, en donde diferentes centros han podido meter datos en una base computarizada, y que esto ha ayudado mucho a tener como homogeneidad de datos, cosa que estamos tratando de implementar con Huntington, pero ha sido un poco más complejo. También en el Instituto Nacional de Neurología y Neurocirugía recientemente hemos estandarizado la evaluación clínica de los pacientes, y básicamente esto es para poder definir cuáles son los caminos multidisciplinarios que nuestros pacientes deben de tener. ¿Cuáles son las barreras y oportunidades que tenemos en México para la enfermedad de Huntington? Todo un poco es redundante, pero estas son las conclusiones de mis pequeños 15 minutos que tengo para hablar de Huntington en México. Básicamente, uno es que no tenemos datos epidemiológicos claros. Todo se basa en los registros de los centros locales. Entonces, va a ser muy importante cuando podamos acceder a estas comunidades y tener las prevalencias reales de nuestros pacientes. Por otro lado, estos clústeres que están identificados en el norte de México actualmente no son accesibles por la violencia y por la inseguridad asociada a los cárteles de drogas. Los datos actuales están limitados a, como mencioné anteriormente, a los centros individuales, y por lo tanto, como no tenemos datos epidemiológicos, proponer políticas públicas al gobierno se vuelve complicado. Entonces, esto sería un buen paso, el cual tenemos que ya arrancar. También tenemos estas disparidades de acceso a salud. Esto es muy importante. México, a pesar de que es un país con muchos recursos, tiene una cantidad enorme de pobreza, y esto es pues muy desafortunado, sobre todo en pacientes que no viven en las zonas más urbanas. Y bueno, no tenemos tanto acceso realmente a las drogas más modernas que pueden ayudar un poco más a los síntomas de los pacientes. Tenemos pocos centros multidisciplinarios de Huntington, y en realidad hay muy pocos especialistas en Huntington, no nada más neurólogos, estoy hablando de neuropsicólogos, de trabajadores sociales, etcétera. Entonces no hay mucha conciencia todavía de esto en la mayoría de los centros de salud, y en realidad en la mayoría de los centros de neurología. Y faltan otro problema es que hay mucha centralización de la atención en las comunidades urbanas. También existen muchas barreras sociales, culturales y religiosas, esto por la heterogeneidad tan grande cultural y étnica que tiene el país, y hay muchos pacientes que por idiosincrasia prefieren no tratarse o prefieren no atenderse de forma occidentalizada, por así llamarlo, para los problemas médicos en nuestro país. Pero no todo está perdido. Existen realmente muchos esfuerzos tanto de la sociedad civil como de los médicos y todos los que nos dedicamos a tratar pacientes con Huntington para poder resolver estas barreras. Entre esto es la creación de estos grupos nuevos multidisciplinarios para Huntington, y básicamente el desarrollo de investigación tanto epidemiológica como clínica en pacientes con Huntington en centros como en donde yo trabajo. Agradezco muchísimo su tiempo y espero que puedan visitar México alguna vez. Es un país muy hermoso y bueno, estoy a sus órdenes cualquier cosa. Muchas gracias.
Hola. Muchísimas gracias, Arturo. Muy buena descripción de la situación en México. Lo que yo invito es a cualquier persona en la audiencia que tenga preguntas para ti, que por favor se aproxime a ti durante los descansos o durante la cena o en cualquier otro momento y ojalá estés dispuesto. Gracias. Bueno, gracias a Arturo, y yo realmente para la siguiente conferencia, para presentar al siguiente conferencista, propuse que simplemente me iba a parar aquí y iba a decir: el siguiente es Bernhard, pero no, Mario me dijo: no, déjame presentarlo bien como se debe y como sea apropiado, entonces Mario lo va a hacer mejor.
Bueno, en eso coincidimos. Creo que el profesor Bernhard no necesita mucha presentación, pero habemos muchas personas de familias y demás, y creo que vale la pena un poco reconocer el trabajo de tan destacado profesional, académico, investigador. El profesor Bernhard LANDWEHRMRYER es actualmente profesor titular de neurología en el Hospital Universitario de Ulm, donde es fundador y miembro del comité ejecutivo de la Red Europea de Enfermedad de Huntington. Él recibió el título de médico y tesis doctoral en la Universidad Albert Ludwig de Freiburg, Alemania. Y bueno, tiene muchos estudios en varias instituciones, en Belfast, Reino Unido, y también en Mass Gen en Harvard Medical School. Y yo creo que el trabajo en Huntington y además el ser uno de los líderes del estudio Enroll HD hace que sea todo un honor el poder contar con su presencia esta tarde. Adelante, profesor LANDWEHRMRYER.
Gracias. So, that's not the first slide. It's a real pleasure to be here, and I really would like to thank you very much for the opportunity to be here. I have to present in English, I'm afraid. I can follow Spanish, but it wouldn't be enough to present. What I'm going to talk about is the clinical trial landscape as it presents to us in 2022. So, before we go, a couple of disclosures. I serve at scientific advisory board, give scientific advice, and here my funding. Thanks. That's the first thing to say. Thanks that I can be part of your meeting here. I learned quite a lot, and I'm impressed what has developed since the last time I was here in 2018. Many centers now in Enroll. I'm really, really pleased that that has happened. What I'm really going to talk about is what we are aiming for. We are aiming for to alter the decline of neuronal function, eventually resulting in neuronal death. And we can do this in a disease like Huntington's disease because we can diagnose it early. And if we were to intervene early, you can hope to modify in a major way the course of Huntington's disease. That's our aspiration. It's the ambition that we are aiming for. It's not an easy thing to achieve, though, given that the pathophysiology of Huntington's disease is complex. We have multiple mechanisms.
Múltiples mecanismos.
Protein
Tenemos la patología
but also other aspects, other protein products being produced. We have alterations at the RNA level. The structure of RNA is different, resulting in different interactions with RNA binding protein, and one of the consequences is missplicing, among others. And at the DNA level, we have a dynamic mutation that is increasing during the course of the life of an HD patient, and that in a region-specific fashion. So that is complex. And we are going to review the complexity of the pathophysiology tomorrow on Friday. What I'm going to talk about now is the clinical trial landscape, and I will parse this into two components. Whoops, that's not the intention. One back if I can. So, I'm first going to talk about ameliorating symptoms and signs, what is new, and then at the efforts aiming at modifying disease progression. I will review with you and for you evidence from completed randomized controlled trials and what we have learned. And I will discuss with you ongoing and planned RCTs in the horizon of the next 2 years or so, and conclude with some comments on the question of hope and hype. If that would advance for me, it would be great. Yes, a word on hype and hope. So, I'm going to focus first on better control of HD-related signs and symptoms and evidence that we have from RCTs here. I will start that off by showing you a patient with a ballistic form of Huntington's disease. She's still able to drink by herself and use cutlery. A year later, you can see that the disease has progressed. The family changed the environment, protecting her, hurting her head with protective gear. She now has to use a straw, and eating is no longer possible with cutlery. So this illustrates that chorea can be pretty disabling and need to be addressed. So one way to think about this that was explored in randomized controlled trials is deep brain stimulation, as we all know, used with quite some success in Parkinson's disease. So what I'm going to show to you is a study, randomized controlled trial, relatively small, 48 subjects recruited. This is not moving the way I would like it to move. Sorry for that. What was used was the Medtronic Activa device, bilaterally, in the region of the junction between external and internal globus pallidus. And the endpoint that was chosen was to compare on stimulation and off stimulation under best medical treatment 12 weeks after surgery. If you look at the baseline characteristics, you can see that pretty clearly. It was well balanced, relatively high total motor score, 55, 56. Let's look at the primary endpoint, that is the total motor score. What you see is that in both the off and the on condition, total motor score is going down with no real difference, no added value, it looks, from the stimulation. Here you see the numbers going down by about 4 points in both groups. If you look at chorea, which was the initial motivation to do the study in the first place, you see a similar picture. You see a decrease in total chorea subscores in both conditions, off and on, and there's no real difference. Here are the numbers. Again, 4 points lower in both groups after 3 months at the 3 months observation point. So the conclusion from that study pretty clearly is that DBS, deep brain stimulation, in the globus pallidus, in HD, is not superior to medical treatment. I've shown to you that there were very variable trajectories of different patients. So some patients may benefit, but it's not possible, at least in this limited small data set, to predict with any degree of certainties who might benefit. It's of course of interest to look at potential explanations, microlesional effects, improvements in both groups observed, bias of blinded investigated, video rating is still out, and Q motor data are still to be analyzed. There are questions about surgical techniques, and then there is a question about the anatomical target. For instance, we observe often in patients who develop subdural hematoma that the first thing that goes away is chorea prior to a clear central paresis. So that's may suggest that there are other regions to consider and other tracts to consider for this. So, the pointer is not working. Randomized controlled trials for symptomatic relief of chorea that are in planning that I would like to introduce to you is a study that, if that would move, we could go on. So, a study that was, the design of which was presented at the recent European HD meeting, a compound called beventolol, a beta blocker, and the mechanism of action is supposed to be a VMAT inhibition. That's not the only compound, as we are all aware. Tetrabenazine, deutetrabenazine, and valbenazine are acting on the same molecular target. The claim is that this particular compound, SOM 3355, may differentiate given that it, from tetrabenazine, given that, wonderful, thank you very much, that it may also act on a VMAT1, not only VMAT2. And we'll see whether that will translate into benefits. It's not entirely, okay. So there are, in a first proof of concept study, some hints that the compound may be active, and there are hints that this is happening in a dose-dependent fashion, and a phase 2b study is currently in planning. So we are hoping that eventually we will have ways to ameliorate hyperkinesias by antichoretic medication without the worsening of bradykinesia and cognition that we often see currently. We are all aware, and we just heard from Saul, that there are other aspects than the motor aspect, behavior we heard from Eric, and cognition from Saul. One important unmet need is to ameliorate cognitive deficits in Huntington's disease. And one approach to this is proposed by SAGE. Again, the presentation of the current state given at the EHDN meeting in Bologna a month ago. So the idea here is that in part, cognitive impairment in Huntington's disease may reflect altered glutamatergic neurotransmission and dysfunction of NMDA receptors. And the compound under study, SAGE-718, is thought to be a positive allosteric modulator at the NMDA receptor. Here is where the compound is supposed to act molecularly. So 2 studies are underway, dimension by SAGE. The primary endpoint is an instrument that has been established for Huntington's disease, although not fully validated and for all languages, on a HD cap. And the study looking at the impact on this primary endpoint is called Dimension, and it's currently done in English-speaking countries only, and is well underway currently. It's obvious that the biggest unmet need is to change the natural history of Huntington's disease. And therefore, let me briefly review with you where we are. In principle, monogenetic disorders could be addressed by correcting the primary disease-causing alteration at the DNA level by various gene editing techniques. Or by replacing, in cases of loss of function mechanism, the faulty gene, or by suppressing the continued production of the detrimental mutant gene in case of a gain of function mechanism that would be reducing gene products in plural at the pre-mRNA, mature RNA, and mRNA and protein level. So there are several tools out there to do so. Antisense oligonucleotides acting at the pre-messenger RNA in the nuclear compartment, splice modulators orally available acting in the same compartment, RNA therapeutics currently mostly delivered by gene therapy, then DNA acting compounds like zinc finger proteins, and then agents acting at the DNA directly. So aside from these targets, I'm really sorry for this pointer here, or the system here, or it's me, I should say. So cellular level pathophysiology, system level pathophysiology are the targets that are important to address, and we will hear about them today, and for instance, directly after my talk, and Michael Hayden is going to talk about predoptin aiming at delay as disability, and there are attempts to restore, interfering with downstream pathophysiology, attempts to restore brain cholesterol metabolism by AskBio, and tomorrow Elena Cattaneo will present her preclinical studies on the very same subject. Let's look at this a little bit. Brain cholesterol metabolism using gene therapy by AskBio again, and data related to the community at the European HD network meeting in Bologna. The idea is that cholesterol is essential for neuronal function, for various cellular functions, and it is known that there are alterations in Huntington's disease. A special focus is on one regulatory enzyme called CYP46A1 for the approach that AskBio is taking. So using a vector And preclinical studies in 2 types of mouse models were conducted, resulting in the development of a gene therapy vector, an AAV10 vector, delivering a cargo intending to influence cholesterol metabolism in the context of Huntington's disease using a stereotactic gene therapy approach. This is the study that is planned for that purpose, a phase 1, phase 2 dose-finding study focusing, for obvious reasons, on safety and tolerability and primary efficacy. Okay, now let's look at evidence from randomized controlled trials, how we are doing concerning the disease modification that we are aiming for through gene silencing. Gene silencing approaches comes in various flavors. Treatment with antisense oligonucleotides, either tominersen non-allele selective, originally developed by an Ionis, the WAVE program with an allele selective approach. Then there is gene therapy approaches potentially with long-lasting effects, focusing on RNA therapeutical agents or zinc finger proteins. And lastly, there's gene silencing with small molecular medication. All of them focusing on transcription on mRNA with the impact on all genes, products, mRNA as well as protein downstream from that. And there's a range of modalities out there to do that. Let's focus on antisense oligonucleotides first. They needed to be chemically modified in such a way, here in violet and in red, that they are lipophilic, so they can pass through lipid bilayers, but they are not able to cross the blood-brain barrier, so they must be applied intrathecally, as we all know, by intrathecal injection. And if you do so, then the nuclear compartment here, the spinal cord, is reached. You will hear a little bit later from Daniel Cirano from Roche in Argentina, giving you an update on the tominersen clinical studies. I will just do a few brief comments on the program. And you all recall that there was a dose-dependent lowering of mutant huntingtin in CSF. However, the dosing in a generation HD1 phase 3 randomized controlled trial was stopped, as we are all aware, at the 69-week point, due to the observation that there was an inferiority, safety concern for that reason. Here in blue, the group with the highest exposure to tominersen. No real difference if you compare the orange and the black lines, that is placebo, and the lower exposure to tominersen. So the question does arise, why did that happen? Why was no clinical benefit of HTT lowering observed? An issue is that there was no formal dose finding or phase 2 study performed for tominersen. So it's possible that there are toxic side effects of an exposure of too high a dosage of tominersen. A possible explanation. It's quite clear that the biological mechanism underlying this toxicity are currently the subject of debate and are not established. I personally favor that it's, in my opinion, likely that an inflammatory response to higher dosages of tominersen or ASO in general may result in ventricular enlargement to disbalance between reabsorption of CSF and production. But that's hypothetical, that's a speculation. It's what we will see, and that will be portrayed to you in the following talks, a planned phase 2 RCT that will hopefully give the answer whether dosage and population is the critical component. And application of tominersen in the context of Huntington's disease. Okay. So you will hear the answers to this. There is a wave approach in allele, in the selective silencing, 2 studies using a single nucleotide polymorphism in the mutant allele of Huntington has been conducted. The SNP1 and SNP2 approach did not result in a lowering of mutant huntingtin in CSF. And what is currently ongoing is a phase 1/2b study using a third SNP. It's called SELECT-HD study. And recently it was reported that there appears to be a reduction of mutant huntingtin with this approach. So let's focus on RNA as therapeutic agent. We will hear from the uniQure approach a little bit later, so I'm not going to focus on this. It's a non-selective approach using AAV5 and is going to target the levels of exon 1 containing the polyglutamine expansion as well. Seotactically applied, you will hear the details in the following talk. Let's then conclude with small molecules, orally available splice modifiers, The idea is that through the orally available splice modifiers, in an intron, normally an intron, an exon is defined, resulting then in degradation of the mRNA with this small molecule defined stop codon in a newly defined exon by nonsense translation-linked mRNA decay. It's a complex mechanism, part of the quality control of an mRNA overall. That results throughout the body and throughout the brain in mutant and wild-type huntingtin lowering, and with 2 types of compounds currently explored here. Novartis showing in children with SMA, for which the compound was originally produced, a reduction, and a pertinent publication explaining the mechanism for those who are interested in the details for it. Currently, the dosing with this compound developed by Novartis is stopped. It's stopped due to emerging safety signal predicted by preclinical tox and axonal polyneuropathy for a thorough review of the data, and we are waiting in the course of this year whether dosing of branaplam can be reinitiated or in which way the trials on branaplam are going to be going forward. PTC is offering a compound that is also orally available and does cross the blood-brain barrier, and thus offering huntingtin lowering both in brain and the periphery. It's in preclinical modeling showing an almost equal effect in the periphery as well as the blood, a similar mechanism that I explained to you earlier. On the intron is identified where the new exon, C exon, is defined. A phase 1 trial in healthy volunteers was completed, and resulting in the conclusion that the compound is well tolerated, and therefore a phase 2 trial is currently underway for a 12-month duration using various concentrations of the compound, with the primary endpoint safety and tolerability and lowering of huntingtin in the blood compartment as well as in the CSF, and with the intention to look at clinical endpoints. However, the primary endpoint is safety and tolerability. So that's the current status of what we have. And let me conclude with a word on hype and hope. There were several setbacks over the past 2 years. Roche announcing that for safety reasons, dosing of tominersen had to be stopped. Wave, with 2 of the explored compounds resulting in no lowering of mutant huntingtin in CSF. Unicure with a higher dose on temporarily on a pausing of the application of higher dose. We will hear about this a little bit later, and Novartis in the Vibrant study talking about safety concerns in the form of a polyneuropathy. So sometimes this may feel like falling, but I guess what counts is that you bounce back. The HD community is used to setbacks, and it's true that not every hopeful wish will translate. But I believe the important thing for all of us is that we can trust in the scientific process that will us to treatments that really work. So the route to success is to go from failure to failure with undiminished enthusiasm. I think it's not a bad saying. And for you in the audience, it's important that you continue to help pushing, particularly in difficult terrain. Thank you. So, sorry for my mishandling advancing the slides. But, well.
I don't think it was entirely your fault. Thank you very much. It was a great talk, and particularly the end is very inspiring, very on point. I think we can allow half a question, if anyone.
Yep.
Beck.
That was a great talk, thank you. A really nice overview. I was wondering, you gave some speculation on the inflammatory process that may have been a reason for the decline in the highest dose for tominersen trial. However, the lower dose, I was wondering if you had the same thought on the lower dose. In other words, did the lower dose also cause inflammation? Because it did not prove an effect in that lower dose.
I think you're making an excellent point that based on the clinical endpoint that I showed you, there was normally somewhat lower on a point estimates for the readouts that I showed you with the lower exposure. However, no statistically significant difference. So you could argue it was not better than placebo, but at least no worse. Remember that the dose of tominersen taken, 120 milligrams, is relatively high. And preclinical studies for a range of ASOs tominersen included, show that in the areas where the CSF is flowing out and where the concentration of the chemically modified ASO is highest, that is spinal nerve roots, there is an inflammatory reaction, not necessarily in the CSF space. So to me, it's conceivable that to various degrees and depending on a range of factors, there is some degree of outflow reabsorption restriction. As potentially a general phenomenon with ASO of this particular chemical nature, it has been observed for numinersen, used for SMA, it has been observed for tofinersen. And so I favor, that's my view, I favor that we may look at a dose-dependent toxicity of the overall chemical entities used in this context, and therefore dose becomes critical. Getting the right dose becomes critical, because in a way what we induce is some extent of normal pressure hydrocephalus, if you like, and with clinical consequences by these types of compounds. So being judicious as to the dose and being judicious as to the population. With age, resilience towards these alterations may decline and becomes an important objective. That needs to be pressure tested. Therefore, I think it's very important that additional studies are conducted to rigorously address these questions.
Thank you.
Yeah.
Mario, maybe a quick question, and
Sure.
Bernhard will give you a quick answer.
Just wonder if you can make a short comment regarding the importance of including non-represented population, both in genetic studies and also participating in clinical trials, like the Latin American population, in terms of benefits for future treatments.
I think that's going to be critical going forward, that there is inclusivity in clinical trials. And I do hope, and tominersen study did make an attempt to go to not only the usual countries, but also Japan, for instance. In Latin America, it was at least Argentina, and that participated. But that is something that needs to be expanded upon as we go forward. No question. I think it's something that will evolve with the higher visibilities of centers and with confidence in the ability of both the patient population as well as the centers to play at level fields, which I have no doubt can be achieved in these future clinical trials.
Thank you very much. Thank you, everyone. Bueno, siguiente conferencista es Michael Hayden, quite known also for the HD community. Dr. Hayden has had a long academic career, mainly at the University of British Columbia, the Molecular and Therapeutic Center of Medicine. And he has been, before Brilenia, at Teva for quite some years, as well, and he has been the co-founder of several enterprises in the pharmaceutical industry. And he will talk about today, of course, about the efforts that Brilenia has been involved in. And thank you very much for coming, and the stage is yours.
Thank you, Roger. And I just want to say what an absolute thrill it is to be here. I have been very moved by what I've seen, and also moved by the incredible commitment of Factor H to doing something for Huntington disease in South America and particularly Latin America. It's been very moving. So I first learned about Barranquilla some time ago, and I learned about it because I heard this amazing woman singing in 2010 at the World Cup in Cape Town, South Africa. Okay, this was Time for Africa, and this was where I first learned about her as I attended the World Cup in South Africa, and this is where it was. And I thought it was appropriate to acknowledge that Shakira first gave me some insight into something about Colombia. I know it's ignorant, but I heard about Barranquilla by looking up where she was born. And I think everybody knows she was born right here. So that's really incredible. I asked some people in Bogota on my way here what they feel about Shakira, and for most young people, she was their idol. And I was told, yes, I know she's got tax issues in Spain, but they've been resolved. She's completely innocent, and the Spanish government is going to pay her back for all the trouble. I didn't know if that was true, but anyway. Okay, so I want to tell you my own story, my story of the mind, of course, which is trying to solve biology in Huntington disease, but also the story of the heart, the story about how Huntington disease captured my interest very early on. And I also, as somebody in my mid-20s, over 40 years ago, I started looking. I was told that Huntington disease did not exist in Africa, and I started seeing families with Huntington disease. And these were families such as these, and similar to many of the stories we've seen and the families we've seen in Colombia and Venezuela, living in poverty, disenfranchised by apartheid, and also disenfranchised by Huntington disease. And for example, this man, this was the first time I'd seen this family living in a shack, and when you lived and went inside next door, there were 2 children with juvenile Huntington disease, and I also saw the high frequency of juvenile HD just outside Cape Town, 10 miles from Cape Town. And then I was invited, because of that, by Marjorie Guthrie. Now we'll see if this pointer works. No, it doesn't, but there's Marjorie. And Marjorie, and this was the whole world working on Huntington disease in 1979. And you won't really recognize me because then I had a different hairstyle. So you can see next to the late, sadly, the late Peter Harper, there's a guy with a big beard and a lot of hair. I know it's hard to believe, but it's actually me. And Nancy Wexley is there next to Eva Gide, MacDonald Critchley, and a whole bunch of folks who really were working. That's 1979. And I became close to Marjorie Guthrie, who founded HDSA. I'm going to tell you a little bit about that. And what I learned from talking from Marjorie, but also visiting and examining 1,000 people with Huntington disease in every single mental hospital in South Africa, recognized Huntington disease, sadly, was alive and well in Africa. And I was inspired by the resilience, the devotion, the incredible courage of these families, but also I realized we had to do something. And that was a long time ago, and it's been a long journey, but that imprinted me early and shaped the directory and trajectory of my own career, and my commitment was made to these families to do whatever I could in an effort to try and solve some of these problems. So Marjorie always used to write Love and Peace. Marjorie actually got me into the United States and was able to escape for all kinds of reasons and ended up. Those days, there was nothing known. And this is an interview that I just did with Norda Guthrie.
Hopeless and helpless. Yeah. Tell us about
Marjorie's daughter.
well, it was a really bad time. My father kind of started
Woody Guthrie's daughter.
manifesting strange behavior in 1950, actually. And it got worse and worse and worse, and people thought he was drinking too much, and there were just all kinds of reasons happening. He was in the Merchant Marines, guys go nuts when they were, you know, just like one thing after the next. And it was very difficult. It's almost like too much to explain. And when my father was finally diagnosed in 1954-55 with Huntington's, the doctors told my mother that it was incurable, nobody knew anything about it, and they told her to tell the children that he had died, that he would be in a hospital sent away somewhere, and to tell us that he was dead. Not only that, but in order to protect her from any financial, because we don't have health care in the United States the way a lot of you guys do, she would be responsible for all of his medical bills. They had to get a divorce in order to get around that. So it was like one bad thing, you know, tell the kids he's dead, get a divorce, put him in a psychiatric ward. He was in a psychiatric ward for 15 years or so with 40 other patients. My mother, I don't know how she kept her head above water. My father passed away in 1967, and she gathered us around the table, the kids. She was running a dancing school all these years. She was the breadwinner in our family because my father was in the hospital for 15 years. So she gathered us around the table, and I remember she wasn't a violent person, but her fist hit the kitchen table for the first time that I ever saw. And she said, "I've got to do something about this. I've got to do something about this." And she looked at all of us. She said, "Are you kids okay? You okay? You okay? You okay?" We all said, "Yeah, Mom, we're okay." She goes, "Okay, I'm going to go do something about this." And she put an ad in the Times, the New York Times. It said, "If anyone knows anything," I'm sorry, if anyone knows anything about Huntington's disease, call VIA 0249.
And that led to the formation of the Huntington Disease Society of America, the first lay patient organization in the world. And then really from there, so many organizations have been modeled and rolled. And we see the amazing situation here in Colombia and Venezuela, also with patient-based organizations. So that gives you some indication of the journey of the heart and the people that had huge influence. Let me tell you more about the science. So of course, as we looked at this, and as you look at this, the key, and you've heard from many speakers today, is of course to have symptomatic therapy, but to try and direct most efforts, and my own methods, have been to see what we could do to try and slow down progression of HD. Now, of course, whilst at Teva we got deutetrabenazine approved, which is great. It's tetrabenazine, but improved. It has less side effects, has had some influence, also used for tardive dyskinesia, and today is actually Teva's best-selling drug. So I want to tell you about pridopidine. And pridopidine is an investigational drug. What we do know so far, it's safe and tolerable. It's taken orally, and that's very helpful, because as we think about the rest of the world, and we think about places where some of the infrastructure and expertise is not as present as it might be in the US or Canada and parts of Europe, an oral drug is very important. We've shown, and I'll give you just some data, that the drug may show some early evidence that the drug may show some maintenance of TFC, total functional capacity. We now understand the biology of the drug. It is a highly selective, specific activator of the sigma-1 receptor, and some really early, very early data, open label, suggests the effects may be as long as 5 years. Now, without spending too much detail, one of the ways you define how a drug has targets in humans is to use radioactive ligands, and what you do is you inject those, you see where the radioactive ligand, this was to the sigma-1 receptor, it binds to the sigma-1 receptor, you see it labeling, then you give your drug. You do another experiment, you give your drug before and you see how much of it is blocked. And what you can see here is almost completely blocked by predopidine, which indicates that certainly this is a major target for this drug. It was also done with targets for the dopamine receptors, D1, D2, and others, using another separate experiment, and there was no binding, there was no diminution of signal using falapride, and so this suggested that this was really predominantly, at this dose, a sigma-1 receptor agonist. Now, what we do know is that sigma-1 represents an intriguing target. It's highly expressed in brain areas that are implicated, basal ganglia. I'll show you how it regulates crucial pathways. We have preclinical data that showed that predopidine actually activates sigma-1. And how do we know that? We know that in all these experiments, repeated many, many times, if you delete sigma-1, you lose all the neuroprotective effects of predopidine, or if you develop an antagonist and use an antagonist. So this tells you from the preclinical perspective that the neuroprotective effects are mediated. So here's the high expression of predopidine in the basal ganglia and the cortex, important areas, of course. When we look at the mechanisms, and this then became a focus for us preclinically to define what mechanisms predopidine is having effects on. So when we look, for example, at axonal transport, mitochondrial function, ER stress, autophagy, BDNF secretion, synaptic function, and these became collaborative experiments and initiatives with about 15 different labs, all trying to assess the role of predopidine in each of these mechanisms, and then looking to see what happens when the animals were crossed to sigma-1 knockouts or a sigma-1 antagonist was used to see what the effect was. So the sigma-1 receptor is a very interesting receptor. It sits on the mitochondrial associated membrane of the endoplasmic reticulum. As you see, it's there. Its normal state is inactive. It's held in an inactive state by an interacting partner called BIP. When an agonist gets there, what happens is that it's released from BIP, it's active, and then it acts as a molecular chaperone acting on numerous targets in different compartments of the cell. It improves mitochondrial function, and I'm not going to spend, these are all published independent data from different labs around the world, so it has improvements of mitochondrial function. It acts on the cytoplasm to enhance autophagy and increase BDNF secretion. It also restores, and very important for ER homeostasis, restores dysregulated calcium levels, has big impact on ER stress as assessed in different readouts, and importantly also plays a role in nuclear cytoplasmic transport in restoring this, in particular certain transcription factors getting into the nucleus that are important and stabilizes the nuclear pore. The end result of all of this is enhanced neuroprotection. And again, when sigma-1 is deleted or inactivated, the effects are completely abolished. I won't go through too much of this, but just to show you, this is work from Chris Ross's lab, where you can see initially here at baseline 0, you can see the cells with the mutation. This is animals with 82q, have cell death about 35%. Then you use predopidine, a certain dose, 1 micromolar, and you see the effect is completely diminished. In other words, it can restore neuronal viability. Then you do the same experiment again, but with these animals now crossed to sigma-1, and you lose the effect completely. This is repeated. This is beautiful work just published from Fred Sadhu's lab, where he looked at essentially BDNF transport using a particular microfluidic chamber that reconstitutes the corticostriatal network. And what he showed is that you could basically restore the corticostriatal pathway, BDN axonal transport was enhanced, and the number of striatal spines was also increased. Increasing spines was also seen by another group, which showed here. Here you can see the wild type at the top. There's a lot of spines. HD spines are diminished. You give predopidine, and it's enhanced. And then you cross it, and that's shown again there. The control is, these are yak transgenic mice, where the spines are decreased. Predopidine in the third purple restores it. You do the same experiment now with predopidine in animals that don't have sigma-1, and you lose the effect completely. I won't go through this, but just to say predopidine also enhances autophagy, and much data to associate with this. So the next effect from preclinical data is it enhances BDNF, increases transport, increased spine density, synaptic function is restored, autophagy is enhanced. All mechanisms that are the consequence of the mutation for Huntington disease and restoring function. So we then embarked in a study whilst I was still at Teva in an effort to see, this was a phase 2 dose-finding study, in an effort to see if there was anything to suggest that this was really a drug that had some effect. Now, initially, when the trial started, this was still thought, the biology was thought to be mainly dopamine modulation. And initially, we planned a 6-month trial in an effort to look at that. The trial failed to reach its endpoint. It failed to reach significance with TMS. And in fact, the placebo group actually did even better than the treated group, essentially at all doses, 45 milligram in particular. But when we looked at this, we had exploratory endpoints in the trial, of which TFC was one of them. And when we looked at it, at the same time, the trial was extended to 12 months because we recognized that this may have a neuroprotective profile, and we would need at least a year in an effort to see effect, for example, on an endpoint as TFC. So the trial went 12 months. This was the 6-month data where the initial endpoint was 26 weeks. We went to 52 weeks, and TMS still showed no real improvement. There was still a massive placebo effect. But what we saw at 52 weeks with total functional capacity, which I think you all recognize, measures those aspects of function that are important to patients: occupation, finances, ability to do domestic chores, activities of daily living, feeding, dressing, eating, going to the washroom, and who takes care of you. And this scale has been around a long time, really 40 years. And of course, for families, this is a very important scale because it measures the activities of daily living, and it essentially, the burden of becoming dependent on others becomes a big deal, and TFC measures those things that are important. This is also a regulatory accepted endpoint, both by EMA and FDA, as a single regulatory endpoint. And these are just notes that talk about it. The EMA talks about meaningfulness, acceptability, TFC, acceptable, and so this became an important exploratory endpoint for us to look at this. And of course, as you all may know, that TFC, the rate of decline is measured annually, so it's a annual scale, and in different stages of disease, this really, it's defined by TFC, there's a different rate of progression. So very early in stages 1 and 2, that would be measured by a TFC greater than 7, the rate is about 0.8 to 1 a year. Here's what we saw. This was the pre-specified exploratory endpoint. I would say very surprising. I didn't expect that patients and subjects who were on the trial would show, and on the drug itself, would show almost no progression. Placebo went down 0.8. Post hoc, because this was pre-specified, post hoc we looked to see what was contributing to this, because in the trial we had about 25% of patients were late-stage HD, and what you can see here, this was primarily driven by patients with early HD. This had never been seen before. There had been no drug that had ever shown any capacity to slow functional in any analysis. So this led us to, and led me to think that this was worthy of a clinical trial. The trial failed. The trial failed because its initial endpoint was TMS, 6-month TMS, and this was exploratory as we went to 1 year. And so, yes, we tried and failed, but I didn't want to fail to try again in an effort with what we had learned right here. And just to show you the TFC, when you looked at all the domains that contributed to the overall finding, it was really all of them except occupation, where patients often in the early phases of the disease will still be working, but had trends as 0.13, but all the others were significant associated with this. So also post-hoc, we also did other analyses, looking, for example, where there were subgroups based on baseline age and CAP score, and the median age in this study was 48, the median CAP was 500, and we did various analyses to this. And what you can see here, that basically in all of these subgroups, we saw the same significance, although mostly seen, for example, in all HD, then low CAP was significant, high age was significant, low age was significant. The high CAP was less significant, but there in each of these were very few numbers, 22 patients, for example, in the high CAP group. So despite these numbers, the TFC effects were statistically seen in 3 or 4 subgroups. So when you're doing post-hoc analysis, you do want to show that the post-hoc analysis gives you some significance in these results. We also did initial analysis. The trial was started in 2004, and filament was not yet wasn't all patients had blood remain, but we did on all that we had. There was no collection of plasma dedicated for this, but we did from all patients from whom plasma was available, and we started doing NFL analyses using Simoa technology. And firstly, we wanted to see whether this followed the trajectory that would be expected for NFL. So for example, here on this is looking in the placebo group. What you can see here is that as the NFL goes up, the TFC goes down, and this is showing going down means going worse. So this exactly what you might expect, that there was an association between decreasing TFC and increase in NFL. This was now corrected for age, CAG, BMI, that can all influence NFL, and this still remained quite significant. When you looked at the 2 groups, we saw that in the treated group, there was some stabilization of NFL. And certainly when you looked at the placebo group, this followed the trajectory seen for numerous other studies in longitudinal studies of NFL levels. So this also, when you just look further in the placebo group, there was a significant correlation between worsening in TFC and increase in NFL. That was significant, but you can see in predopidine itself, it never went above 0.5, and this was always associated with stabilization for TFC. So it was this data, suggestive data, that suggested that it was worth getting in the ring again, going for a few more rounds, going to see whether there was any chance that this drug actually really had an effect in a randomized clinical trial with TFC as the primary endpoint in an effort to really assess that particular parameter. We also looked, by the way, the convergence of data also suggested that there might be a beneficial effect on motor and behavioral measures. These were all post-hoc assessment to try and generate additional confidence that it was worth spending the next 5 years doing this trial. Now, Q-motor, we also looked at. Q-motor is more sensitive. It's rater independent, it's been used a lot, it's non-invasive, read centrally, and so we were very interested. We had Q-motor as part of the study, and so this was more sensitive. And here you can see the Q-motor data. It's not published, but here you can see, looking at inter-onset interval, various ways of relating that. There's certainly placebo, for example, went down as we normally see. So this is quite different to the TMS data, where in fact the placebo and the treated group, placebo actually did better than the treated group. But in Q-motor, we actually started seeing some difference between the 45 milligram group and the placebo. And of course, Ralph Riemann has shown a very nice correlation looking at composite data between Q-motor and TFC. This all fell together. We looked at other measures, none of which really reached significant, but all had trends in the right direction. We've heard about apathy. Well, what you can see here, placebo went down as expected, and that decrease was mitigated with predopidine, small numbers, 59, 62. Not powered for this, all post hoc, but all looking to see if there was anything else we could believe. And when we looked at the improvements in which symptoms, there was apathy, disoriented behavior, irritability, all going in the right direction. That gave us, again, some additional confidence. The other issue that really became also quite compelling is that there was quite a lot of data also showing that this was safe and tolerable. And that also, then, the side effect profile in prior studies had shown a side effect profile very similar to placebo. And these are just some of these prior to this study. These are looking at PRIDE, MERMAID, and HEART, and you can see the frequency of these side effects, no real significant difference from placebo with drug itself. This is an oral drug, and particularly as we look at places outside of North America and parts of Europe where the infrastructure is not there, the expertise not there to do, for example, intrathecal or even intracerebral injections. So we really would like to see an oral drug forming some basis for therapy, its ability to reach its targets, its ability to be administered easily, and particularly a drug that has a low side effect profile. So let me share with you where we are. This is a little out of date, October the 7th, but close. So firstly, despite COVID, we were very worried that we would not be able to roll. It's 60 sites around the world, 30 in North America and Canada, 30 in 9 countries in Europe. We enrolled 6 weeks ahead of schedule. That just talks about the Huntington community and their willingness to be partners in an effort to try and develop different approaches to therapy. We're on track to read out in the second quarter of 2023. We powered this on a 20% dropout rate, but the dropout rate so far has been 7%. There will be more dropouts, but it's not likely to be 20%. What's also the case is that now about 60 people who have been eligible, have reached the end, have gone into an open label part of the study, and 100% of people have done that. It's been assessed by our safety board, who have felt there's no safety signals of concern and recommended the study should continue. There have been numerous serious adverse events, as would be expected in any trial, but none have been deemed related to study drugs. So overall, this is encouraging in terms of safety and tolerability. Here you are, the study, the endpoint is the change in baseline, change from baseline in TFC, a whole host of other endpoints. It's important to note that with the new, I would say, really landmark staging classification, the international system for staging, that all these patients are in stage 3, early stage 3, and I would say that new approach to staging is landmark, is going to be very catalytic for all trials going forward, but we can now see that we have patients that are really all in stage 3 in that particular trial, in this trial. So here we had a common close design, which is, just to explain that what is that until the last patient has had their last visit, everybody stays blinded until 78 weeks, and then we go into open label. So what you can see here is that we expect to have 450 patients, more or less, who complete the endpoint at 65 weeks, but we will also have additional data on 334, or around that, we now know, who complete 78 weeks in a blinded manner. So we'll have data at 52 weeks, 65 weeks, and 78 in an effort to assess safety and tolerability and durability of efficacy of the trial. The FDA has granted fast track for us. We're having more frequent meetings with the FDA, and these have been extremely collaborative, cooperative. They've accepted essentially the full stats plan, and everything's on track. Just to say, there's another trial also that's ongoing right now that is just finished, because this drug was also chosen by Mass General and Merit Sutkovitz for the Healy platform trial. And this was because the safety tolerability was recognized, but also there was unique validation because the mutations in sigma-1 cause ALS. And so and furthermore, there's a genotype-phenotype correlation. So if you have a complete mutation and no protein, you have onset in childhood, you have a partial missense mutation producing a dysfunctional protein, onset in adulthood. And this trial is now has come to the end with the fourth arm of this platform trial. Sadly, all three prior drugs as part of this have failed. One of the reasons perhaps is that this trial had patients who had 1 to more than 3 years onset of their duration, so very late onset. And I would say, have a very low likelihood of success with pridopidine because probably the patient inclusion criteria were very broad, and pridopidine, there was no powering against any endpoint in ALS. So despite all this encouraging data, it's important to remember that this is a phase 3 study. 40% of phase 3 studies fail for all kinds of reasons. 60% are successful. So we do not go into any of these results with any greater likelihood of success than 60%. It's not bad, but it's certainly not a sure thing at all. So these are the endpoints we're reading out in second quarter 23 for Huntington's. We will have the results of ALS in about a month from now, and we'll learn from that, I'm sure. And it is important to know, where do we want to be in Huntington disease? Our own view is that Huntington disease is likely, in the end, if you want to stop or slow it, to require a cocktail of drugs. We would love to have a drug that deals with the cause, but even by the time you deal with the cause of Huntington's, secondary pathways will already be abnormal. You'll need more than a single cause, unless you're going to treat people very, very young. So it's likely to require a cocktail of drugs, just like many other diseases, like cancer, other neurodegenerative diseases. It's not going to need a single magic bullet that's going to solve this problem. The problem at the moment, we're dealing with sick care, not health care. We're treating patients who have disease. As other people have said, ideally, you'd love to treat people before they have disease. Delay onset, keep people active, independent, loving their children and grandchildren, walking, exercising for as long as possible. Any delay would be fantastic. And in the end, we'd love to find ways to reverse or repair HD. So in conclusion, from this discussion today that we've heard, it is an exciting time in HD neurotherapeutics. I like to think of it as a brave neuro world, because that's where we are. It needs bravery. The drug pipeline in HD is growing. There are numerous people coming into it. And the whole field has learned so much from failure. Nobody's, as Bernhard so beautifully said, nobody's just lying down with this. We're picking ourselves up, and we find something to learn and keep on going forward. You, the patient community, are deeply engaged. And we believe that, and I believe that this will require an approach which includes different targeted treatments. And the long-term goals are to prevent and reverse HD. So finally, I just want to say, this is Woody Guthrie. his writings became a deep inspiration for me through Marjorie and through Nora and Arlo. And Woody, despite all what he was going through, and also a fighter for amazing social justice, spoke about the fact that you should not let any earthly calamity knock your dreamer or your hoping machine. And Woody Guthrie, who was the really inspiration for Bob Dylan and so many other, and really the founder of folk music in the United States, I think there's a better world coming. And let's listen to what Woody sang about that just to end this off. We just got...
Why, there's a better world that's coming, I'll tell you why. There's a better world that's coming, I'll tell you why. There's a better world that's coming, I'll tell you why.
So I think there's a better world coming when we look at the pipeline of Huntington disease drugs. We don't know when, but it is surely coming.
Thank you very much, Michael. It was a very nice talk, also a very nice introduction and a very nice end of the talk. 2 half questions. If someone. In any case, if you come up with any later on, just please feel free to reach to Michael. Thank you very much again. Let me hold on to that. And please stay in your seats for a few more seconds. We will have something before we continue with Daniel Ciriano from Roche.
Hola. Explícales en español y te van a traducir.
Okay. Hola, soy Leila Mieles de Factor H, Colombia. Todos los que estamos aquí somos programa Abrazos, todos los chicos de Factor H. Está Zuleima, Arnel, y están todos los que faltan. Estamos aquí hoy, está Atlántico, Piojó, Saco, Vaivén, Juan de Acosta, Barranquilla, Algarrobo, que es Magdalena, San Ángel, Difícil, vino Riguacha, que es La Guajira, vino Cesar, que es Valledupar. Y les queríamos dar gracias a todos por estar aquí, porque sabemos que somos importantes para ustedes y porque mírennos, somos felices y nos reímos por ustedes.